Good morning, everyone. We will be starting shortly. We're just waiting for a couple more attendees to pop in. So as soon as we are full house, then we'll get started just shortly after 9. We'll be on mute until then, so we will start the webinar in a couple of minutes. Thank you so much. Okay, I think we'll get started. Kathy, you can hear me?
Yes, perfect. Thanks, Robin.
Yes, wonderful. Right. Welcome, everyone, and thank you for joining us. Some of us in the morning, some of us in the afternoons. Today's topic will be touching around BioGX's latest portfolio of lyophilized real-time syndromic multiplexes for research use only. It will be presented by Dr. Katarina Stein, which I'm sure most of you are very well affair with, our product support manager at BioGX. Just a couple of items before we get started. All attendees will be muted throughout the webinar, so no one will be able to chat with us openly across the board. However, if you refer to the left of your screen, you will see a little Q&A icon, a little question mark. If you have any questions, please feel free to pop them in there. Once the webinar has been fulfilled, Kathy will then go through those questions. Those that we don't have time to answer, we will send to you in an email in a question and answer format, along with the recording of the webinar. So with no further ado, Kathy, the floor is all yours.
Perfect. Thanks, Robin. Thanks for the introduction. I'm going to share my screen. So hopefully this will all come up for you without problems. Is this all okay, Robin? Can you see this? Can you see the screen, Robin?
Yes.
Perfect. Great. So yeah, let's get started. I'd like to welcome you also to today's webinar, and thank you very much for joining. And as Robin mentioned, this presentation will provide you with an introduction to our new product line, which are the real-time syndromic multiplexes, or so-called research use only products. And throughout the presentation, I will refer to the products as research use only products, but on our website, they are listed as real-time syndromic multiplexes. I will give a brief overview about BioGX and what we do as a company, just for everyone here who is not yet very familiar with us. I will talk about our diagnostic product range, and then we will dive into our newest product line, which are the research use only products. Okay, so starting off with some company highlights. So we are one of the pioneers of developing and manufacturing fully lyophilized diagnostic assays. We have started doing this in 2007 and have built up our experience ever since. We are a fully integrated global business, which means that we control all processes from product innovation to to research and development, to manufacturing, and also to product delivery. And the manufacturing and R&D side of things, they all happen in our headquarters in Birmingham, Alabama, in the United States. And then we have our subsidiary company here in Amsterdam, in the Netherlands, from where we distribute our products all around the world. And when the COVID-19 pandemic hit early last year, we made sure that our supply chains and manufacturing infrastructure remains intact, and we can now produce 5 to 8 million tests per week. We've also created this sample to answer portfolio for COVID-19, and we currently have those 3 assays that are listed here on the market. So first of all, the SARS-CoV-2 assay for the BD-MAX. Then we have our combo assay, which detects SARS-CoV-2, flu, and RSV, also for the BD-MAX. And then our extraction-free assay for COVID-19 as well. We have strong industry partners. So we work with, for example, Becton Dickinson, we work with Bosch, Thermo Fisher, and we use Medline as our distributor in the United States. And then we use our around 40 global distributors to sell our products into more than 80 countries globally. All of our products are provided in our lyophilized sample-ready format. And for this, the only step that's needed is the addition of water or rehydration buffer, and you're ready to go, and the reagents are ready for use. So the lyophilized format brings multiple advantages. So for example, all products are shipped at ambient temperature, which means there's no need for cold chain shipment, so no need for wet ice or dry ice. And there's also no freezing or thawing of the reagents. And the lyophilized format simply makes the product very reliable and reproducible as well. And because there's minimal preparation steps involved, this also means that there's no loss or waste of any materials. And it also makes it less prone to operator errors because there's very little handling steps involved. This slide here, we really wanted to highlight our testing library. So on the slide, you can see the main molecular diagnostic companies down in the bottom and the number of relevant syndromic targets that the company can detect with their assays. So the targets are divided into different groups. So for example, the blue one represents the respiratory group, red is for STIs, and yellow is, for example, for UTIs or women's health. So we currently have assays on the market that cover 218 targets, which you can see in the little black box on the left. And we have also further assays in the pipeline that will cover another 186 targets, which means that we are in direct competition with Seegene. And it's also worth mentioning at this point that we provide template positive controls for all of our targets that we can detect with our assays. So some of our targets might be unique and it might be difficult to source any control materials. And our template controls, they are also provided in our lyophilized format. And they have a concentration of 100,000 copies per lyophilized bead. And for DNA assays, the target sequence is cloned into a plasmid, which is then lyophilized. And for the RNA assays, it's a single-stranded RNA that is also lyophilized. The template controls also just need to be rehydrated with water, and then they are ready for use. And customers find them handy, for example, for daily run controls, or they use them as to generate standard curves as well. Okay, so our products really fall under three major market segments. And our current product lines cover the clinical diagnostic segment, the non-clinical, and the pharma QC segment. The non-clinical products focus on food safety and water quality, and the pharma QC includes an assay for the detection of heparin source contamination. But really, the clinical diagnostic product range is our main focus. So we offer a wide range of clinical diagnostic tests. So we provide, for example, open system reagents or contract specified reagents in the United States, which are basically customized assays that are developed and optimized from us for our customers. And then we have assays on the market that are EUA or FDA EUA approved. And we have our CE-IVD and laboratory use only products that you might be more familiar with. And the newest addition to our product line are our research use only products. So we'll talk about those during this webinar. And the difference for the RUO products is as well that they are for distribution and sale on a global scale. The RUO products will follow the format of our previous assays. So the main product features are that all assays are provided as sample-ready lyophilized assay. And with the release of the RUO products, we will have about 60 additional assays that will be available. And in total, they will cover more than 200 individual infectious disease targets. And this gives the customer the flexibility to run one extract of one patient sample on different tests to get different results at the same time. The new feature of the RUO product line is that is the availability to serve different PCR platforms in addition to the BDMAX. And this is facilitated by the variety of lyophilized formats that we offer. For the products that are for multi-platform use, we have primers and probes incorporated that detect an endogenous sample control, which is the RNase-P. And then for RNA assays, we'll also have an internal amplification control which is a single-stranded RNA. For the BDMAX configuration, we'll have primers and probes included that will detect the regular sample processing control that some of you already might be familiar with. And again, as for the other products, shipment takes place at ambient temperature. There's no need for dry ice or wet ice. And also the lyophilized reagents contribute to the long-term shelf life stability. So the PCR platforms that are compatible with the REOs will be the BDMAX, as already mentioned. And then this product line can also be run on the applied biosystems, so the ABI Constudio 5, the ABI 7500 FastDX, and the two Biorad systems, the CFX 96 Touch and the CFX 384 Touch. Of course, the REO products are compatible with other platforms that we have not validated or that are not listed here. So all the PCR platforms that have compatible optical channels can be used. And if a customer wishes to validate different platforms, we are of course happy to provide technical support and assistance if that is needed. The next couple of slides, we really wanted to kind of highlight the syndromic groups that we target and all the different microorganisms that we can detect with our assays, just to showcase the wide range of different targets that we include in our assays. So just to note, so basically everything that's labeled here in red, this means this is a new target that has not been included in our other assays. So this is a new addition in the REO product line. So when you, for example, look at the blood syndromic group, we have a couple of bacterial targets here that we've added. For COVID-19, we will be able to detect the E gene as well of SARS-CoV-2. For the gastrointestinal syndromic group, there's multiple additions. So you can see, for example, Campylobacter, Salmonella, Shigella, and so on. When we look at the immunocompromised meningitis and encephalitis group, you can see that there's 3 viral targets that we've added. For the respiratory group, we've added adenovirus, coccidioides, and JC virus. And for STIs, we'll added the Gardnerella vaginalis into it. For the UTIs and the wound syndromic group, we have a broad range of new targets that we included here. So again, you can see here, for example, Acinetobacter, Clostridium, Enterococcus, Staphylococcus as well. And then for women's health, we have 4 additional bacterial targets as well. So going back to the multi-platform compatibility that we already talked on a little bit before. So all the REO products will be available in 2 formats. So one is to be used on the BDMAX, and the second one is to be used with the other PCR platforms that we touched on as well. And the products that are compatible with the BDMAX, it's indicated in the lot number. So they will be called 450-series-max, just to differentiate them from the other product configuration. And for the BDMAX configuration, one kit will consist of 2 different items. So there will be one pouch that includes our lyophilized PCR master mix, and then the second pouch will hold our rehydration buffer. One kit is sufficient to test 24 samples, and materials that are required to perform the test but are not included in the kit are the extraction kits manufactured by Becton Dickinson. These are specific for the BDMAX and have to be purchased separately. The same for the PCR cartridges that are to be used with the BDMAX. And the tiny tubes at the top, you can see this is what it looks like. So this is the snap-in tubes for the BDMAX extraction kit. So on the left, it includes the lyophilized pellet, and on the right, you can see it holds the rehydration buffer. And in the bottom, it's just what the pouch looks like. The version that is compatible with the other PCR platforms have an LMP at the end of the lot number. And for this configuration, the kit consists of one item, which is just one pouch that has the lyophilized PCR reagents. So they are provided in 2 8-tube PCR strips. And you can see a picture of it at the top there on the right. And for this, there's also a few additional consumables required that are not provided in the kit. So the customer needs to purchase the PCR instrument compatible consumables. So that's really important. And also molecular grade water is needed to rehydrate the pellet or the lyophilized master mix. I also wanted to talk about this multi-platform compatible configuration in a little bit more detail, just to give you some more information and to explain the differences. So we have multiplex assays included in the RUOs that detect 2, 3, or 4 different targets. And those assays make use of 4 different optical channels. So we have the FAM, Hex, Texas Red, and Sci5 equivalent optical channels that are needed to perform the test. And then the remaining multiplex assays, they detect 5 different targets. So they make use of an additional optical channel, which is the Sci 5.5 channel. And you can see examples of compatible PCR platforms that are listed below the tables. So again, these are the platforms that we have validated, but of course there's more platforms out there. So if they have the optical channel compatibility, they can certainly also be used. The next 2 slides, we'll talk about the sample processing workflow just very briefly, just to give you an idea what it looks like when the customer has to set up the samples using our reagents. So just as a recap, so for the BDMAX compatible product range, the 450 series Max, so the PCR reagents are provided in sample-ready lyophilized format. 1 kit is sufficient for 24 samples. It's to be used with the BD extraction kits and PCR cartridges purchased separately from BD. And the time to result for this is approximately 2 hours, depending on the PCR profile that is being used. And on the top, you can see on the left what the PCR cartridge looks like. And then on the right, just a schematic representation of the extraction strip. So the first step is really to load the BD extraction strip with our PCR reagents and rehydration buffer. So the 2 snap-in tubes that I showed you on a slide earlier, they are just snapped into the extraction strip. The sample is then added to the sample buffer tube. The sample buffer tube comes with the extraction kit. And then the end user loads the PCR cartridge onto the BDMAX instrument. And then the sample buffer tubes that now includes the sample and the extraction kits are just loaded onto the BDMAX platform, and the run is started. So that's the sample processing workflow for the BDMAX. For the multi-platform assays or product series, the sample processing workflow looks a little bit different. Again, as a recap, PCR reagents are provided in lyophilized format. 1 kit is sufficient for 64 samples, and this is to be used with silica column or magnetic bead-based extraction kits, and molecular grade water is needed to rehydrate the lyophilized master mix. The time to result is approximately or is less than 80 minutes, again, depending a little bit on the PCR profile that is being used. And just to highlight here again that it's really important to know that the 8-tube strips that we provide that hold our reagents, they're not compatible with, for example, the ABI QuantStudio 5, with the ABI 7500 Fast DX, or the 2 Biorad platforms here. So the customer really needs to purchase the PCR consumables that are compatible with the platform the customer is using on site. Regarding the sample processing workflow, so the lyophilized master mix is provided in those 8-tube PCR strips. The user adds 40 microliters of molecular grade water into 1 tube to rehydrate the master mix simply by pipetting up and down a couple of times. And then 10 microliter of the rehydrated master mix is dispensed into fresh PCR tubes. And of one rehydrated PCR tube, 4 new PCR tubes can be filled with 10 microliter of master mix. Then the extracted patient sample is added, so 5 microliter of the extract is added to the rehydrated dispensed master mix. You can give that a quick spin just to remove all the droplets from the top of the lid, and then the PCR can commence. Yeah, so to wrap it all up and just to kind of touch on the key features of the RUO products. So for the validation, so the validation is done by the customer, and this is required for RUO and LUO products. All of our CE-IVD products are now available as research use only products. And just to remind you that the manufacturing and also the QC processes are identical for the CE-IVDs and the RUOs, so there's absolutely no difference. The RUO products simply provide more flexibility, for example, when it comes to product registrations that might be very thorough in some countries and might be very time consuming. So the RUO products could be used as an alternative in those cases where CE-IVD product registration is just very time consuming. We have all the product inserts for the RUOs readily available to download from the website. And also the BDMAX UDP files, the run files for the ABI and the Biorad platforms, they are available upon request. And then we just go through the website navigation just to show you how you will get those documents and this information. So you'll go to our main web page. You hover over products, and then this little drop-down menu comes down, and you click on real-time PCR syndromic multiplexes. A new window opens up, which just shows you the MAX and the LMP configurations and just tells you a little bit about the different configurations. If you scroll down, you can see the different syndromic multiplex groups that we cover. If you scroll down a little bit more, you can see the different syndromic groups just with the individual RUO products that fall under the different groups. And if you then click on, for example, this is the Plasmodium assay, if you click on the little plus symbol, another kind of drop-down box opens up, and with a little description or a short description about the assay. And then you click on product insert, and this is what the product insert will look like. And the product inserts, as I said, are available to download for all of the RUO products. Okay, so this was everything about the RUOs. So you've all received our updated product catalog and price list recently, and we would like to take this opportunity to also tell you a little bit more about this new real-time PCR platform called the Pixel 16. And we are very pleased to announce the release of this portable PCR system. So it's a small footprint instrument, and you can see. So basically, the lid is the same size as an Apple iPad, so it's very small. And it really supports the testing capacity through decentralization and point of care applications. And initially, we are planning to launch this platform coupled with the extraction-free COVID-19 test. And here are some key features of the Pixel 16. So it has 16 wells, so 16 samples can be tested at once. It has 4 optical channels, so up to 4 different targets per well can be detected. The instrument is operated through the touchscreen that is integrated in the lid of the instrument. And also the amplification can be monitored in real time on this screen while the run is ongoing. There's no need for calibration of the instrument, so it can be used right away. It uses Android software, and also it has LIMS connectivity. Another great feature is that it has a battery backup option. So in case there is a power cut or it needs to be used outdoors, this is also possible. Yes, and another thing to mention is it has an open system or it is an open system platform, which means that the user can also use it for other PCR runs. Yeah, with that, I would like to thank you very much for joining us today. And I hope the presentation about the RUOs was helpful. And as Robin mentioned at the beginning, we'll be happy to answer some of your questions now that came through the Q&A function. So let's take a look at those questions.
Kathy, I think you can start with the one towards the bottom, which says, is there a difference between LUO and RUO products?
Okay, perfect. Yes. Yeah, so there's actually no difference. So it's just a different terminology. So I think RUO is just more widely used, and to avoid any confusion, we decided to use this term going forward. So LEO stands for laboratory use only, and RUO is research use only. Okay, I see another question coming in, and this one is, will performance data be added to the RUO product inserts? So no, this will actually not be the case, because for research use only products, we will not provide any performance validation data such as limit of detection, inclusivity, clinical performance data. So this will not be provided in the product insert. And as I mentioned before, for the RUO products, the customer has to perform the validation on site. Another question is, why aren't LUO products on the marketing documents? So, the LUO products that we currently have available, so they will remain available for purchase to customers, but we will not expand the LUO product line. So everything going forward will be RUOs. So the RUO product line will be expanded rather than the LUO product line. Okay.
Okay, if there aren't any other questions coming through, give everyone a second to see if they want to put anything else in there. I see one or two others, Kathy.
Yes.
Mm-hmm.
So I think another question is, are you planning to CE mark the RUO products? So I think this has to be reviewed kind of on a product basis, depending on the market demands and the forecasts. So certainly if there is a big demand for an RUO product, this can be discussed. Right now, we don't have any plans to CE-IVD mark the RUO products that we have released, but this can certainly change in the future. Okay, I think this is it. And as Robin said, all the questions that we have not answered here, we will provide them in an email or with a follow-up after this webinar.
Wonderful. Thank you, everyone, for joining us this morning. We really appreciate you taking the time to listen about our new portfolio. If you have any questions, please feel free to reach out to us. After this webinar, you will receive the full recording, as well as any questions that weren't answered or that we've missed in this lineup, because there's quite a few coming in at this point in time. So we thank you very much, and hope to see you all again soon. We will be signing off now, and please once again reach out if you have any further questions. Have a lovely day. Thank you, Kathy.
Thanks, Robin. Bye.
Bye-bye.